We can see.
Oh, okay.
Yeah.
All right. Hello, everyone. Thank you for organizing this meeting. Thank you for the opportunity to present. And today I'm going to talk about computational approaches for discovering and studying somatic mutations. So, but I will start with more like general introduction. So we as mammals, we develop from a single fertilized egg, which is shown here. And then the development follows very defined process. So first there is the formation of blastocyst, then blastocyst, cells in the blastocyst separate into inner cell mass and trophoblast. Trophoblast will eventually make up most of the placenta, and inner cell mass has a further differentiation into epiblast, hypoblast, which then differentiate into germ layers through process of gastrulation, and then cells in the germ layers will start form organs. So, and this process is very reproducible, and it's kind of very deterministic. So if we look into more detail, so this is how cell differentiation roughly looks like. This is a compilation of different studies, and actually it was published over 10 years ago, but maybe this can be updated, but overall it gives you overview and understanding what's going on. So this is a fertilized egg, and this is how cell lineages develop in our body during development. So for example, this is blood lineage, this is brain lineage. So this picture, excuse me, it shows you differentiation in terms of cell specialization, how cells decide who they're going to be and what they're going to do in our body. At the same time, so this process of differentiation is coupled to the process of acquiring mutations. So these processes go kind of hand by hand in development. And then, so what can happen, you can have mutation at any stage of development, actually from the very beginning, also from the right after the fertilization. And then if mutation happens relatively late, let's say in the blood lineage, it will be present in a few cells, in some cells of blood lineage, but can be completely absent everywhere else. Or it could be the way around. It could happen relatively late in the brain, and then maybe have some phenotypic and harmful consequences, but completely undetectable in blood. So, and actually can be even situation if mutation happened relatively early, like soon after the fertilization. So it will be present in brain and blood cells, not in all of them, in some fraction, and can be even passed to the next generation, to the germline lineage. So, and then this is developmental process, but then in aging also, we can have acquired mutations. So this is that accumulation of somatic mutation and how they're related to health in our lives. This is a major point of interest in our lab, and this is what we are studying. But my lab is purely computational, so we develop a lot of bioinformatics approaches and collaborate with experimental labs to design experiments and generate data and validations. So a little bit about mosaicism. So what is the definition? So mosaicism is the presence of two or more derived from the same cell, but genetically distinct cell lines or lineages in the human body. So it's not a chimerism. Chimerism is when you have different genomic background and they mix within one body. Like the simplest example of chimerism, this is when you transplant organ to a different person. So now you have organ with one genetic background and the rest of the body was different. So we're talking about here that actually cells have different genome, but they are ancestrally related. They originated from the same zygote. That's important to understand. In fact, this is an old term. Idea that different cells within one body can have mutation was expressed in 1904. The term somatic mosaicism was first time used in 1940 for pigeons. And actually the first mentioning that there is somatic mosaicism in human, specifically in this content, was done in 1945. But recently, there is a huge interest in somatic mosaicism because with development of technologies, we understand that actually widespread and present in all of us and actually in every single cell of us. And then now we have ability to study it not just in a limited cases, but basically systematically and at a single cell level. So there could be people with different background. I just give a quick overview. What do we mean by genomic variants? So oftentimes people mean just a point mutations, but actually it's more than that. So in our genome, we have two haplotypes, paternal, maternal, and they're actually not necessarily identical. There could be some differences. This is genomic variants. This is and these are a few examples. One of them is single nucleotide variant. There is a difference only in one base. So there could be few bases consecutively different. So this is called indel. This is abbreviation from insertion and deletion. And then there is actually, when it's relatively small, it's called indel, but when it's relatively large, it's called structural variation. So for example, structural variation here, this is tandem duplication. So one haplotype has just one copy, and there are two consecutive copies on the other haplotype. There could be like similar like this, there could be deletion. So this definition by 50 bases is relatively arbitrary. Sometimes people use different, but the point is that it's not only a point mutation. You can have relatively large chunks of DNA which are altered. So then we also have mobile elements which can copy and paste themselves in our genome, and actually quite large fraction of our genome consists of these mobile jumping elements. So if it's inserted in one haplotype and the other haplotype doesn't have it, then it will look like this. So for this haplotype, it will be a deletion. For this haplotype, it will be insertion. Then I already talked about structural variants. This is tandem duplication. There could be deletion like shown here. There could be inversion of the sequencing. And actually, there could be events which are complex. So they can combine different flavors of everything what is shown here. So you can have a structural variant, which is not simple deletion. So it could be deletion, duplication, plus some indels, plus point mutations around. So all of that. So, but one important thing to understand that currently the way bioinformatics works, all the interpretation and all the data analysis are done relative to the reference genome. So reference is being constantly updated, but it's really not that important how it's updated. The conceptually important to understand when we are saying there is a, let's say, mutation or deletion or duplication, we all the time mean it's relative to the reference, typically. So for example, in case here, so if there are two haplotypes in this person, and there is a and T here, and the reference has A, then in such case, we will say that variant is T, or mutation is T. So here, in this case, if there is a CTG, then in the personal haplotype, we will say there is a deletion on, in personal genome, we will say there is a deletion over here, and so on. So that's an important thing to understand. All right, so how can we study somatic mosaicism? This area is actually booming and quickly changing. So there are many approaches developing now and new technologies. But if you kind of go on our literature and see what people do, so effectively there are two general approaches. One of them is you take a bulk of sample and you just try to analyze that bulk. So here is some sample or tissue, and I use circles to represent cells, and different color means that there are different mutations. So you take this bulk, you generate typically sequencing data, everyone works with sequencing, and then you generate prediction, what are the somatic mutations in this bulk. And then obviously as a good scientist, you may want to do some validation, and then you can probably use the same sample. So in this case, we call it technical biological validation. In a second, you will understand what's the difference. So that's approach number one. So approach number two, when you have a sample, you will say, well, bulk is, it's kind of like a soup of everything. I want to be more specific. Why don't I analyze the mutations at a single cell level? So you'll take a cell and you'll try to you extract the cell and you try to get DNA out of it and generate the data. So the problem here is that the amount of DNA in a single cell is really small. It's a 6 picogram. And almost no current technologies can work with this amount of data. The only one I know is a fish, which can give you reliable estimates for some copy number and big rearrangement, but this is really huge rearrangement. If you want to go to like more detailed analysis, you will have to increase the quality of the DNA, the quantity of DNA, and you have to multiply DNA somehow. So you can do it by two ways. So there are enzymatic ways to amplify genome, which is called whole genome amplification, or you can take a cell and grow a colony out of it. So, and then once you have a basically once you clone a cell and then you get DNA, so from DNA generate data, and then you can compare it to the DNA from the bulk, compare the data, make calls for the mutations. And then here there is a significant distinction in terms of validations. So if you are using DNA that you amplified for validating your calls, we call it technical validation. If you use DNA extracted from the original sample, we call it biological validation. And what is the difference here? The difference that this DNA here and DNA from the bulk are different in this step where you do cloning or amplification. So during that step, you actually may introduce variations in your DNA just by chance, like experimental error. So you start manipulating with the sample. And that's an important distinction that exists between these two validations. That's why we call one technical, biological. So, and this step, it's kind of like, as I told you, inevitable, and then have to be really careful when interpreting your results with the technical, biological. What is the limitation of technical validation? So in principle, you can maybe validate all of your calls and say, well, I have like 100% sensitivity and specificity, but that may reflect some of the artifacts that you introduced during generating the DNA. If you do biological validation, the problem is here that kind of you are free of this amplification step, but the limitation is that some mutations, which are very rare, you may not be able to ever validate them simply because they're extremely rare. In extreme case, as it's shown here, if you have mutation present in just one single cell, you took the cell, you clone it, or you amplified it, then this mutation is not present anymore in the bulk, and you will never be able to validate it. So that's a fundamental limitations of these approaches. So, and the difference between analyzing single cell and bulk, probably clear to you by now, that in a bulk, you can only find relatively high frequency mutations, like it is shown here for cells in blue. And in a single cell strategy, theoretically, you can find any mutations, but the question comes of the false positive, which you may not be distinguishing, or sensitivity when you may not be validating all of your variants. So, but both approaches used by different strategies, and in fact, they're actually complementary. Because when you do bulk, you can find frequent mutations, but when you do single cell analysis, in order to find frequent mutations, you can actually need to sample a relatively large number of cells, and that becomes expensive. The other distinction is that whole genome amplification actually doesn't work that well yet. We don't have very reliable technologies. So recently on the market coming new approaches, which seems to be way better than before. But so far, the considered the gold standard approach for single cell analysis is cloning. So I'll start from analysis. Oh, sorry, this is not showing you correctly. I will start with a single cell analysis. So none of the project, what we were interested to study mosaicism of the fetal brain. So we extracted cells. This is post-mortem samples from abortion. So we have brain samples from the fetus, and cells are still live. So we can actually, if you have cells, so you can, and they have different mutation, you can clone them, get these colonies, and discover mutations. So of course, the question was, how do we discover mutations? And that may seem apparent to you, but it is not, for the reason is that if we have mutations which are present in a bulk, like let's say in a tissue, and is present in a single cell, so when you compare single cell to a bulk, it's hard to distinguish. Is it really present in this? If it's present in both samples, it's very difficult to make a judgment. Is it really a mutation, or is it really, or maybe it is a germline variant which was inherited from parents? So that's a question we try to address, and in fact, it's actually a very important question. So the way we try to address it is to use a strategy when we are not comparing single cell to a bulk sample, but we are comparing single cells, it's clones. But clones is a proxy to single cell. When we compare clones to each other. So here's an example. So let's say you have a tissue and you have relatively frequent variant in the tissue, relatively frequent mutation. Then comparison of clones 1 and 5, which have this mutation, to the tissue will not identify you because it will look like it's present in both of the samples. Instead, what we use approach that we have multiple clones, we compare them to each other. And for a real mosaic somatic mutation, so comparison of clone 1 to clone 2, 3, and 4 will identify it as a mutation. Similarly, for clone 5, we will also identify it. But when we compare 1 to 5, we won't find any differences. So there will be no hit for this mutation. And there will be no differences when we compare other clones. So we will get this matrix of hits. So if we have a germline variant, which is present in all of the cells, so then comparing clones will not show us anything. So we will have this, but it's not going to supposed to show us anything. But occasionally we'll have a situation when one cell somehow lost this variant, and then we will get some hits, but these hits will be arranged vertically in the table. And finally, if we really in supposition we're not supposed to find any variant, it's not present there, but we can have just natural noise, but noise will be sporadic and will look something like this. So now, if you look at this, the way that hits are arranged in a table for real mutation mosaic, for the germline and false positive, they're quite different, and then you can computationally resolve this issue. So we conducted this analysis initially in the brain sample. This is a rough result. So this was a 3 fetuses that we have 3 brains, and every dot represents you a single clone, and our y-axis is a number of SNVs. And this analysis shows that number of point mutation, it actually increases with age, and this is 15 weeks post-conception, this is 21 weeks. And we estimated that it increases on a rough rate of 5 mutations per day, but we have relatively large statistical error. So the range is from 1.6 to 20 new SNVs per cell per division. So it's actually quite high rate, but it's consistent with the previous estimate, which gave between 1 to 3. So there is an overlap. So in fact, it actually shows that we accumulate a lot of mutation, our cells accumulate. Basically, every single mutation we typically get, every single division will typically lead to one mutation in our cells. And you can imagine that we have trillions of cells when we are born. So we have trillions and trillions of mutations. So a little bit more about importance of this approach. So this is another study. We did this actually living person. So we took fibroblast biopsies. From fibroblast, we can derive induced pluripotent stem cells. These are clonal cells lines. And so again, we compare clones, and then we discovered mutation. But what we can do from this analysis, by using mutations which are shared between different cells, we could reconstruct ancestry. So this is shown on the next slide. Here. So so these are so over here on the right. So these are individual cells which were cloned via IPS lines, and then the letters defining mutations. So for example, this mutation A, B, C, D. So they are shared between all of the cells, and this mutation like alpha, beta, He, delta, and so on. So they are shared by this cell. So there are 2 groups of cells. These cells and these cells don't share any mutations. So which actually means that this group has common ancestor and this group have common ancestor, but they don't have any common mutation between them. So that makes us believe that actually there should be a common ancestor for all of them, because we know there should be in our body. So then our hypothesis or interpretation of this, that this is a zygote, and we can actually reconstruct using mutations, retrospectively reconstruct what was the first division during the history of this particular individual. So using this mutation which happened early during development, we can actually see the individual history of development for a person. It's kind of like a personal cell ancestry rather than like, many people use ancestry to define what was their relatives in the past. So here you can kind of define what was common between cells within one single person. And we can now do it in every single individual. All right, so just moving on. So a little bit about the second strategy. So sorry, somehow my arrows are switched by mistake. So talking a little bit about bulk. So we were part of the Brain Somatic Mosaicism Network. This project existed for about 7 years, and it was completed last year. And then, so what we did, we sequenced the brains to high depths. And then there were multiple diseases, and I will focus on 3 of them with whole genome sequencing and about point mutations. So the challenge was when we started how to discover somatic mutations. Tools existed to discover mutations which are germline and maybe somatic mutation in cancer, but here we're talking about just normal somatic mutations which occur during our lifetime. So what we did, we took genomes of 4 independent people who were well characterized and sequenced before, so they're completely unrelated. So, and we mix them in different proportion. So now the germline variations, which are shared between 2 or a few of this genome, they will give us some allele frequency. And those which are unique will give us a low allele frequency. So at the end, we simulated this kind of distribution of allele frequencies in the mix. So there was mix 1, so these are proportions here, the mix 2 are proportions here. And then if you combine mix 1 plus mix 2, then you get slightly different proportions. So, but anyways, it's actually, we physically mix cells to simulate a perfect experiment where you get a sample, and then we have all these allele frequencies. So now we can take the sample of mixed cells, you can sequence it, and you already know what you're supposed to find, and you can test the method development. So when we did this initial experiment and use different programs, so this is the same as I showed before here. So what we find out that existing tools really gave us strongly discordant results. We had only 1 point mutation, which was uncommon. Oh, sorry, this is actually a real sample. No, please ignore this. But we actually had a lot of discordance, and we looked at the sensitivity, and sensitivity was very different. So with existing tools back then, we typically couldn't discover anything below 10%. And if we could discover it, this discovery was not reliable for low frequency mutations. So we need to develop a new approach. So, and we spend a lot of time on it. So I will kind of not gonna go into details what exactly we did all of this, but I will try to focus on the results. So at the end, we selected 400 mutations from the real sample after we developed kind of best practices approach to discover mutations. And then we did validation for the 400 sites. So eventually, a lot of them were deemed false positive. Some of them were deemed true mutations, and some were germline, meaning they were inherited, they were not somatic. And for some of them, we didn't have enough data. So, so each column here represents a mutation, and each row here represents a different data set. And this is from one single brain, which we used like as a reference for all our analysis and testing. So what you can see is that if there is a real mutation, so color, by the way, represents a low frequency. So if there is a real mutation, it's very reproducible from different data replicates. So this is very nice. But if you see a false positive, surprisingly, it could also be reproducible. And also, more than that, so these are samples which are completely unrelated from different databases. You can see the same false positive in a different samples. So this is important to realize. Reproducibility doesn't necessarily mean that you're getting the true answer, because you can reproduce a false positive. So, and so one of the important things that you can use a panel of normals, so we call it PON, panel of normal genome, which are not genomes like cancer genome or any particular disease, where if you find a somatic mutation in a sample, it typically should be unique, maybe unique on Earth. So, and then you shouldn't find it anywhere else in the normal genomes which was sequenced before. If you see it there, then it's indication this is probably false positive. So this is actually a very powerful filter, how to get rid of false positive. And there were a few other things which I can highlight. For example, typically false positive were close to the homopolymer and STR. STR is short tandem repeats. This is a problem where reads from the sequencing are not aligning well. So at the end, after going through this validation exercise and adjusting our pipeline, we arrive to the following strategies. So what we do, we take a sample, which is sequenced to the high depths, and we use a program, GATK. People who work with variants know that this is pretty standard program. However, a default mode of GATK assumes that there are only two haplotypes. In our case, we set it to the mode that it expects there are 50 haplotypes. So which means that each mutation could be at relatively small frequency for the coverage. So then we remove a good portion of the genome where existing short reads don't work well. So about 25% of our genome is extremely repetitive. It's a lot of repeats or very long repeats. So unfortunately, in these areas, we completely give up. So we only leave at about 75% of the genome where we can work. So we filter out mutations, variations which exist in population. We require that they're going to be significantly different from 50% in a person. And then we use a panel of normal filter, which is shown here. And at the end, so this actually took us a while to figure out that even with all of these filtering that we use, so you still have to use, you still get relatively high amount of false positives. So we use machine learning approach. So one of the machine learning approaches shown here, mosaic forecast, which actually uses many features to figure out whether the somatic mutation is false positive or true variant, and that actually made final filter and arrive to the goal to give us best practices to arrive to a good call set. So testing testing this call set on somatic mutations, on simulated mutations. So we actually see that we get pretty good sensitivity. So this is a different modes of running best practices. So now we can, our sensitivity is around 70%, and it's quite high, down to a frequency of about 2%. So that we have a little bit limitation here in the area where it is high frequency, because high frequency now become indistinguishable from the inherited mutations. And then based on extensive validation, so we validated 43 somatic SNVs in a reference brain. We estimated that the sensitivity is roughly 90% in the accessible genome. Accessible, this is where we don't count repeats. And it's about 65% across entire genome. And as I said previously, about 25% of loss is just simply because we throw away the difficult part of the genome. Okay, so now we have this very great tool. We actually can now take sample, sequence into high depths, and discover somatic mutations. And don't be surprised that this is only 43, because somatic mutations, although I said they're quite common per cell level, those which are very common and present in many cells, they're actually relatively rare and much smaller than the variations inherited from parents. So what did we find? So this is an analysis of all the data. I don't go much into detail, just give you some small preview. So this is the coverage we had per sample. So we had 3 different cohorts, Tourette syndrome, autism syndrome, and schizophrenia, and then we had normal brains. And they were sequenced to roughly the same coverage, between 200 to 300x. And this is number of mutations that we discovered per brain. So this is in the log scale. So you can see that typically what we discovered, it was about 20 to 60 mutations in a brain, somatic. However, in some brains, we discover a much higher number of mutations, particularly this one. It was close to 2,000 mutations. We actually, with more thorough analysis, we showed that this brain likely had a cancer, but it was undiagnosed. And this is all postmortem brains. I forgot to mention this. This is all postmortem. And cause of death was typically some accident. So probably this person likely had a cancer, but it was undiagnosed, and we can now diagnose it basically on molecular level on mutations. So a few things which are demonstrating here that we get very consistent results in terms of mutation spectrum across all of the brains. And this is a quality control. When we phase mutation to haplotypes, we have limited power with short reads, but we can phase about 20% of mutation. That they all show that actually majority of the sample is very consistent. So the blue bar means it's good. So red bar means it's not so good, but it's just two samples which have a quality issue with the data. They're a little bit outlier, but overall we get very consistent result, reliable call set. And if we compare mutations that we discovered from this analysis with those we saw in the fetal brain, which I mentioned a few slides above, that we can get very consistent mutation spectrum between them. So it's great. So now we have a tool. We can actually study it not only in brain, but in other tissues, see what somatic mutations there. So just a few slides left. So I didn't go too much into details what we did with this mutation, but I'm going to show you a few more things, what we did in the brain somatic mosaicism network. So we also analyzed copy number variations. So the difference in copy number variation is the following, just to emphasize. So point mutation is just one base has been changed between the haplotypes. So in copy number variations, what we have, big changes. So it's either big region has been deleted or maybe duplicated. So when we sequence with the reads, so the copy number variation can be discovered by the decrease in the read depth. So we will have just fewer reads mapping to this area of the genome. And if there is a duplication, there will be more reads mapping to the genome. So this is how we discover. So we specifically developed tool CNV Piter to discover somatic copy number variations. So before we had a tool CNV-nator, which was oriented on germline, and CNV Piter is successor and also expanding CNV-nator to do mutation discovery, somatic mutation, somatic copy number. So just briefly what it shows, it's use read depth information, which I already mentioned to you, but it also looks at imbalance of the haplotypes. So let me go back for 1 second. So here, if you start, if you suddenly delete one haplotype or duplicate one haplotype, now you have an imbalance. And so natural variations within these areas start becoming an imbalance. So here they are balanced, they are 50-50 two haplotypes. Now you see imbalance, so you can use this information. It's called B allele frequency. So what this approach does, of course, we do a lot of normalization, but the ultimate key feature is that we have these two signals together and we segment them in two-dimensional space. So we have one space for the read depth, one space for the B allele frequency. And when we do segmentation and trying to find copy number changes, we use two signals at the same time, which buys you power, statistical power. So what did we find? So we developed this method, we tested it when we applied it to the brains in the brain somatic mosaicism network. We had roughly around 200 brains. So this is one of the examples where we can find copy number variations. So this is a region of cortex, and this is a region of striatum. So this is coordinate on chromosome 3. So, and this is a copy number. So typical average copy number is 2. So you can see that, yeah, so here, copy number 0, 1, 2, 3. So if we would have a full duplication of the haplotype and inherited from parents, the increase of the duplication should be up to 3. If we would have deletion, it will go down to 1. So what you see here is that it's actually increased, but it's not increased up to 3. So it increased a little bit. So this is clearly a somatic duplication. And then the same happens in the B allele frequency. So if it's perfectly balanced two haplotypes, it should be 50. But if one haplotype dominates on the other one, so you'll start seeing imbalance in terms of frequency for regular SNPs inside of this duplication, and actually we see the split. And interestingly enough, so this duplication present in the cortex, and it's also present in striatum. This is quite far away regions in the brain. And we also can see it in a different cell fraction we extracted from here. So it's very consistent. We actually can identify precise breakpoints where this duplication happened. You can run PCR across breakpoints. You can validate them. So it all works out. So our interpretation of this, because it's present in two brain regions, so very likely that this is CNVs that happened during development. And as I said, maybe you remember one of the first slides. If it happens relatively early, it will present across multiple cell types and tissues. And that's our interpretation here. It's one of the development. And typically we find only developmental CNV in about 1 out of 15 brains. So, on the call we have, I think, around over 35 people I see right now. So I guess in 2 of us, we can definitely, we're likely to find CNVs. So this is showing here what is the length of the CNVs, what is their cell frequency. So we typically find CNVs which are present in at least 10% to 30% of cells, and size varies largely. So the smallest one we could find is below 100 kb, in size, and the largest one, it was loss of chromosome Y, which was actually observed in the blood very often, but now we see it also in brain. So despite the fact that this is only 1 out of 15 brains we can find copy number variation, because of their size, that may be the largest somatic diversity that exists in our genome. However, still we only find 7 cases, so I guess larger sample size is needed to make confident statement. So with this, let me acknowledge my colleagues. So my lab is computational. I have many people involved in this project. So they're listed here. We interact a lot with the lab of Flora Vakarina from Yale University. This is people from her. And then also we interact with lab of Alexander Urban from Stanford. And many other people contributed to different parts of work, particularly in the BSMN project. And of course, the BSMN project itself. Thank you very much. And thank you for your attention. I'll be happy to answer questions. I hope I'm on time.
Thank you very much for your interesting presentation. We have one question from Dr. D. In clinical practice, they usually look at things like compare normal tissue versus tumoral cells on the genes of concern per patient for somatic versus germline discrimination. So, do you mind explaining the downsides of the basic clinical method and as your separation of germline versus somatic mutations achieves more important insights? Could that be done per patient or this a population based for your work?
Oh, okay. So, right. In clinical cases, like in cancer, typically what people do is they compare cancer genome versus some normal tissue. And I think for cancer, it makes sense because what cancer by itself, they are clones, right? So there is a one single cell which actually expanded, and then what matters is everything what happened during this expansion. So, and that works well. But everything what happened, but things which are common between normal tissue and cancer will never be identified. So that's a big limitation. And the question is, do you want to identify such things? In cancer, maybe not, but in some other cases, like if you're looking for some other diseases, and particularly if you have a disease where there is no clonal expansion expected, like let's say something happened in somatic mosaicism mutation, which can lead to bad phenotype. So then comparison of tissues will be actually kind of have very reduced power. So it's likely mutations are not spread evenly across of our body. We know that. So you can have cases sometimes when you take sample, you find developmental mutation in one sample and you don't see it in the other one. So in such case, when you compare 2 of them, you will be able to find this mutation, but there is no guarantee. In fact, we don't know exactly how mutations spread in our body, but we know that they are not evenly spread. So, and that's why, yeah, it could be that if you're really interested in somatic mutation and you suspect the somatic mutation can cause disease phenotype, then pairwise comparison of samples will be very limited. So it's better to use a single cell comparison. Though I would say clinically it's still a little bit expensive, but I think that many approaches are being developed now who eventually I think will be applied to clinic and will be usable for clinic. I hope that answers your question, but I'll be happy to clarify if not.
No, I think that was very helpful. Just to hear from different insights. Just brainstorming because I've been thinking about this, and you pointed out a couple of things about MSI, but like chromosomal instability is a propagator for some of these things. What is your intuition on that? Intuition of the the spread of this either cancer or not cancer, whatever we're looking at in terms of differentiation.
Well, I think that this hasn't been well studied, but I think that there is an actual negative selection even within our body. So some cases, so there is a negative and also positive. So it's actually evolution happens not only on the scale of generation. I think it also happens within our own body. A great example of this is a clonal hematopoiesis in the blood. Of course, blood is the easiest to study. So people know that there are some mutations that lead to proliferative advantage, and then you actually have a clone which over age takes over the blood. So in young people, actually, you're not able to see pretty much any separate clones, but in aging people, like almost all of them, you can clearly see that now there is some clone, or maybe sometimes couple clones that dominate blood formation. And then this is example of positive selection, but I'm quite sure that like microsatellite instability, if it happens, right, that probably will be under strong negative selection, because then you're likely to disrupt cell function, and this will be disadvantageous.
Mm-hmm.
But eventually, probably you can find this combination where the rearrangement in the genome will actually lead to proliferative advantage, and then eventually leads to cancer. So in one of our cases that it's not yet published because we don't know how to exactly publish. So we have many IPS lines, and we actually see recurrently CNV that happens in one particular place. And we can show that this is not the same CNV. So it's not clonal. So it's not happened once and then we have multiple cells ancestry, but it happened multiple times. It's not exactly the same, but it's roughly in the same position. So which does suggest that maybe this is some kind of fragile region in this particular person. We didn't see it in other IPS lines from other people, but in one person we actually see that. So that probably exists, but we don't know exactly what it means. So we don't have good interpretation for that.
I don't know if this is helpful, but again, just as from my cancer background, is so some of the places that we look at is where DNA repair is failing. So that would be another way, you know, like weaknesses. So when they do MSI, they've actually identified the regions where they're looking for these copy number variations and where these DNA repairs are not working well. So that's just a thought, if that helped.
Right. Yep. Thank you.
You're welcome. It was a great talk. I learned a lot. Appreciate it.
Thank you. Yeah, I'm glad to hear that.
Thank you. Thank you to all the participants and speakers for coming to this conference. We are happy to announce that our next set of webinars are scheduled on October 2023. Wish to see you all at IGSMP International. Welcoming you all to our sixth set of webinars. Thank you and good luck.
Thank you. Bye.
Bye-bye.
Bye. Thank you.
Goodbye.